This conference will now be recorded.
Hello students. Now we are in the 8th chapter, Cell, the Unit of Life. According to NEET syllabus, in this chapter you are learning about cell, about prokaryotic cell, eukaryotic cell, then plant cell, animal cell, then the cell organelles. About all this we are learning. So here, components of cell, we have started learning about that. So in the components of cell, components of a cell. In that, actually, we know that the cell, if we are saying about a cell, we can say about the outer covering, right? The outer covering, we know that the cell wall and cell membrane. Cell wall and the cell membrane, they are the outer coverings. Then we learned about protoplasm. Protoplasm, that is the fluid content of the cell. Then the cell organelles. Right. So like this, we are learning about a cell. So in that, the cell organelles, we have learned about the outer covering, the cell wall and the cell membrane. Then the protoplasm, which is the fluid content of the cell, we have already covered. Then about the cell organelles. Right. So cell organelles, in the sense, the structures which are floating within the protoplasm, like mitochondria, chloroplast, ribosome, all these things. So in that, then we are learning the function of cell wall, cell membrane, protoplasm, everything we are learning, right? So we have learned protoplasm. Then about cell organelles. When we have learned about cell organelles, a few cell organelles we have already covered, like mitochondria. Then functions of mitochondria we have learned, the plastids, then functions of that, then endoplasmic reticulum, Golgi bodies, lysosome. About all these things we have learned, right? So, and cell organelles, their functions also we have learned, right? Now we need to move to the next cell organelle, which is ribosomes. Ribosomes are very important. Ribosomes. We have already told that there are some cell organelles which doesn't have a membrane. So here, ribosomes, they are non-membranous cell organelles. Non-membranous cell organelles, and these are like granules. They are like granules. Okay. And who discovered this ribosomes? Palladay. He discovered in 1955. In 1955. In which one? In animal cell. In animal cell. And what about plant cell? In plant cell, Robinson and Brown. Robinson and Brown, they discovered in plant cell. Okay. And in 1958, this term ribosome that was introduced by Roberts. Okay, so who coined or who introduced that time the term uh, ribosome? Roberts. And here we need to mention one very important point, like this ribosomes that is found in both prokaryotic and eukaryotic cell. It's present both in prokaryotes, prokaryotic cell, and eukaryotic cell. Okay, in prokaryotic as well as eukaryotic cell, you can see ribosomes, and in a eukaryotic cell, ribosomes will be freely present in the cytoplasm in eukaryotes. It's free in the cytoplasm. It will be freely present in the cytoplasm. In which one? In eukaryotic cell. So how it will be present in the cytoplasm? It will be attached to freely, in the sense it will be attached to what? It will be attached to the surface of endoplasmic reticulum. So because of the presence of this ribosomes, endoplasmic reticulum, you can see rough endoplasmic reticulum and smooth endoplasmic reticulum. Some endoplasmic reticulum, in some organism, endoplasmic reticulum surface will be having ribosomes. Okay, so those are called as rough endoplasmic reticulum. Okay, and smooth endoplasmic reticulum, they doesn't have this ribosomes on the surface. So here that may be attached to the surface of endoplasmic reticulum or in the matrix of organelles like mitochondria, chloroplast, and these are also found in the membrane uh, which is attached to the nuclear membrane. Okay, so they can also found attached to the nuclear membrane and inside the nucleolus. Okay, and the main point, like when we are explaining about ribosomes, you have seen the structure of ribosome. Each ribosome that consists of two subunits. Ribosome that is having two subunits. Which are the two subunits? The subunits that will occur separately in the cytoplasm, and that is joined to form a ribosome particle only at the time of protein synthesis. So, okay, so that is having two subunits, and these subunits will be present in a separate form. Okay, and only in the time of protein synthesis, only at the time of protein synthesis, you can see these subunits will join together and they will work together. It will join and work together for protein synthesis. Clear? So each ribosome is having two subunits. So the subunits, they will occur separately in the cytoplasm. So they'll be present in the cytoplasm, but both the subunits won't be together. That will be in a separate, that will be there as separate units. And these subunits will join to form a ribosome particle, a complete ribosome particle, only at the time of protein synthesis. So it will be like this, one big subunit and one small subunit. So these will get separated always, or it will be in a separate it will work as separate unit, and only during protein synthesis it will join together and it will work together. Okay, and in prokaryotes, this is one important question. In prokaryotes, you can see 70S type of ribosome, and here the subunits. 70S type of ribosome you can see in prokaryotes. In that, small subunit will be 30S and the large subunit will be 50S. So this question, each type, see, I'm going to explain 2-3 types here. And in eukaryotic cytosol, okay, eukaryotic cytosol, in that you can see 80S ribosome, and here small subunit will be 40S and large subunit will be 60S. Okay, then in mitochondria you can see 55S. Total, or the ribosome type is 55S. In that, small subunit is 30S and larger subunit will be 40S. Then in chloroplast, we can see ribosome type, that is 70S ribosomes you can see in this, and the small subunit or the smaller unit will be 33S and the larger subunit will be 50S. So this you need to keep in mind the types, like you may get a question, the larger subunit of uh, mitochondrial ribosome, that is, you'll get several options. So in that, which option is correct? 40S, the largest subunit. This is smaller subunit and this is the largest subunit. Then we said only during protein synthesis this both subunits will join together and it will work for protein synthesis. So the two subunits of ribosomes, they are associated with the help of Mg2 plus ions. Okay, two subunits. Now we have seen several subunits of each type of ribosome, right? So these two subunits are joined with the help of Mg2 plus ions, right? So they are associated with the help of Mg2 plus ions, and if conditions are there, if Mg2 plus or magnesium ion concentration is less in the cytoplasm, or Mg2 plus ion concentration is less in cytoplasm, these two subunits that will get separated. Okay, in accurate concentration of Mg2 plus or magnesium ion concentration is perfect, then these two subunits of ribosomes will associate together and they will work for protein synthesis. Okay, if the Mg2 plus ion concentration or magnesium ion concentration is less in cytoplasm, both the subunits will get separated, and if the Mg2 plus ion concentration is increased 10 times, Mg2 plus ion concentration is increased 10 times, then what will happen? If increased concentration condition, what will happen? That ribosomes unit that form a dimer, okay, ribosome unit forms a dimer which is of 120S. Okay, then these ribosomes are called as organelles within an organelle. Within an organelle. Okay, and ribosomes, the chemical composition that we need to know. So the ribosomes are chemically composed of rRNA and proteins. Ribosomes are composed of ribosomal RNA and proteins. Okay, and here lipids are absent. Lipids are absent in ribosome, and ribosomes, we know they are the sites of protein synthesis. That means translation, protein synthesis. That process is called as translation. So they are commonly described as the protein factories of the cell. This is the ribosomes are the site of protein synthesis. Translation is the process of protein synthesis. So these ribosomes are called as protein factories of the cells. They are called as protein factories because protein manufacturing will be taking place there, right? Formation of protein. In eukaryotes, the site of synthesis of most ribosomal RNA is the nucleolus. Okay, so in eukaryotes, in the case of eukaryotes, ribosomal RNA or rRNA that will be in nucleolus. Okay, so these many points, ribosome is a very important part. So you should know that each ribosome is having two subunits, and these subunits will be in a separated form. And whenever they need to synthesize what protein, that will join together and that will work together for the protein synthesis, and the protein synthesis process that is called as translation. And here ribosomes, they are called as organelles within an organelle, and lipids are absent in ribosome, and so they are called, they are the site for protein synthesis. So they are called as protein factories. And when these subunits, we said both the subunits are getting joined together. So with the help of this Mg2 plus ions, these subunits are able to join. And when this Mg2 plus or magnesium ion concentration that is less in cytoplasm, two subunits will get separated. And if Mg2 plus or magnesium ion concentration is increased 10 times, what will happen? The ribosome units that will form a dimer of 120s. Okay, so these points you must remember. You can expect question from this area. Then we said different types of ribosomes like 70s, 80s, 55s, 70s. So in prokaryote, 70s type of ribosome. In eukaryotic cytosol, 80s ribosome. Mitochondria, 55s, and chloroplast, 70s ribosome. Then we need to know the subunits, larger subunits and smaller subunit of these ribosomes. Okay. Next, we are going to learn about vacuole. Vacuoles. Okay. Vacuoles, they are the single membrane bound structures. Okay. These are found in where it is present. It is present in cytoplasm, and they are having single membrane. Single membrane bound structures which are found in the cytoplasm. And this vacuoles contain water, then excretory products, sap, then other materials. Other materials in the sense that materials which are not useful for the cell. So that need to be discarded, right? So for that, it is taking the help of vacuole. And all these are like water, excretory products, then sap, other materials, all the other waste materials or the unnecessary materials for the cell. All these are bound in a single membrane. Okay, so we said the single membrane, and this single membrane, inside this single membrane, all these unbonded materials, then excretory products, sap, water, everything will be there. This single membrane is called as tonoplast. Okay, so vacuole membrane, the membrane of vacuole, that is called as tonoplast. And we said the vacuole that will be occupying 90% of the volume of a cell in a plant cell. In plant cell, 90% of cell volume that is occupied by vacuole. Vacuole. Okay. In plant vacuole, you can see the anthocyanin and anthocyanins. Both are found in the plant vacuole. Anthocyanin and anthocyanins. Both are found in plant vacuoles. In prokaryotes, you can see gas vacuole also. In prokaryotes. Okay. So here, which are the main points we need to learn about vacuoles? So it is present in the cytoplasm. It is found in cytoplasm and it is having a single membrane, which is, that membrane is called as tonoplast. So in this tonoplast, it contain, inside the tonoplast, it is having or it is containing water, so the sap, then other materials, the waste materials, then the excretory products, everything will be present inside this tonoplast, okay, which is the single membrane. Then in plant cell, you can see large vacuoles. Large vacuoles, you can see, while learning about plant and animal cell, the difference, while saying about that, we mentioned this. In plant cell, you can see large vacuoles. That means 90% of the cell volume is occupied by this vacuole. And in plant vacuole, you can see the pigments like anthocyanin, anthocyanin and all. In prokaryotes, you can see gas vacuoles. Clear? Next is microbodies. Okay, microbodies. Micro means minute or very small bodies. Okay, so these are small cell organelles. Microbodies, as the name indicates, here you can see they are small cell organelles. Okay, and these are also having single membrane bound. It's having single membrane and this absorbs molecular oxygen. Molecular oxygen. Okay, and that can take part in oxidation. Oxidation. Okay, and these microbodies are of two types. Which are the two types of microbodies? Two types of microbodies are there: peroxisomes and glyoxysomes. Peroxisomes, then glyoxysomes. Okay, see, peroxysides, they were discovered by Dedew. And peroxisomes are also single membrane bound organelles. Single membrane bound organelles. And here the presence of a large quantity of one enzyme, which is called as catalase. So that will convert the toxic hydrogen peroxide into harmless water and oxygen. Okay. So here enzyme, catalase enzyme that will be present in large quantities. In which one? In peroxisome. So this catalase enzyme will help to convert hydrogen peroxide, which is toxic. So this toxic hydrogen peroxide will get converted into what? Water and oxygen. Okay, they'll get converted into harmless water and oxygen. What about glyoxysomes? What and all we need to say about glyoxysomes? This is discovered by beavers. Okay, and where he found this? In the endosperm of germinating seeds. Endosperm of germinating seeds. Okay, especially these are found in fat rich plant cells. Found in fat rich plant cells. And glyoxysomes, they are involved in the metabolism of fat. Involved in fat metabolism. Okay. So how it is metabolizing this fat? Through glyoxylate cycle. Glyoxylate cycle. Okay. So this is glyoxysomes. They were discovered by beavers, and it is seen in the endosperm of germinating seeds, and especially they are found in fat-rich plant cells and fat metabolism. In that they are involved, and fat metabolism is done through glyoxylate cycle. Okay. So we have learned about microbodies, which are the small organelles which is having single membrane and that is absorbing molecular oxygen, and that is taking part in the oxidation. And we need to learn about two types of microbodies, peroxisomes and glyoxysomes. Clear? Next is about cytoskeleton. See, cytoskeleton, they are fibers or cytoskeletal fibers. Cytoskeletal fibers, and this include microtubules, intermediate filaments and microfilaments. Microfilaments, tubules, then intermediate. And this microtubules, they are hollow and cylindrical structures, and that is made up of, or that is uh, built from tubulin protein. Tubulin protein. So that microtubules are built in tubulin protein. Then During mitosis, it is having role. During mitosis, this mitotic spindle that is involved in separating the replicated chromosome. Mitotic spindle helps in the separation of chromosomes. Right. During cell division or mitosis, this mitotic spindle formation will take place, and those spindle apparatus that will help in the separation of that replicated chromosome during mitosis. Okay. So that mitotic spindle, that is the assembly of microtubules. Mitotic spindle, that is the assembly of microtubules. Clear? And this microtubules that is having many functions like chromosomal movement, then intercellular transport. Microtubules, they have several functions like chromosomal movement, then intracellular transport of material. Intracellular transport. For that also it is helping. Then cellular motility. Flagellar movement, ciliary movement, and all those within the cell. Okay, so that is about cytoskeleton. See, these only important points I'm mentioning here. Cytoskeleton or cytoskeletal fibers, which include microfilaments, intermediate filaments, and tubules. So these tubules, they are made up of tubulin protein. Then it is having this microtubules, they are having very good role in the mitosis for the separation of chromosomes. So that mitotic spindle, which is helping in the separation of chromosomes, it is made up of tubulin protein or microtubules. Okay, and that is having some more functions like cellular motility, intracellular transport, chromosomal movement. For all these, microtubules are helping. Then we need to say about cilia and flagella. Cilia and flagella. Cilia and flagella, they are hair-like outgrowths, right? Hair or thread-like outgrowth. And this will be in a 9+2 pattern. Okay. And cilia, we can say cilia are very small. They are small structures. They are causing the movement, or they are helping for the movement of either the cell or the surrounding fluid. Small structures helps for the movement of that cell or the surrounding fluid. And flagella means they are comparatively longer, and flagella, that is compared to cilia, they are longer, and these are mentioned for cellular movement. In cilia, the cell or the surrounding fluid also may move. it may help for that. But here in flagella, they are comparatively longer and they are responsible for the cell movement. Okay, the prokaryotic bacteria also have flagella, but that is structurally different from that of eukaryotic flagella. Okay, prokaryotic flagella or prokaryotic bacteria is also having flagella, but that structure is entirely different from that eukaryotic flagella. And cilium and flagellum, they are covered with plasma membrane. Cilia and flagella, these are covered with plasma membrane. Okay, so that's about cilia and flagella. Next, about centrosome. See, centrosome, that is cell organelle which is having two cylindrical structures called centrioles. This is having two cylindrical structures called centrioles. Okay, so both the centrioles in a centrosome, that will lie perpendicular to each other. Okay, so that organization is like a cartwheel, and these centrosome or the centrioles in a centrosome, they are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Okay, here also the tubulin protein you can see. And the center part of the proximal region of the centriole, that is also proteinaceous. Okay, and that central part, that is called as hub. Okay, and this is connected with the tubules of the peripheral triplet by radial spogs, which is made up of protein. Okay, and these centrioles that will form the basal body of cilia and flagella and spindle fibers, which is giving rise to spindle apparatus. So centrioles, from that is the basal body of cilia and flagella and spindle fibers. See, while learning about the cell separation and all, we'll draw the spindle structure, right? So from this, there is a disc-like structure. From that, the spindles are forming, like that we will show, right? So that structure from that basal body of cilia or flagella or spindle fibers, that is giving rise to a spindle apparatus during the cell division of animal cells or plant cell. During the division of cell division, this is taking place. Okay, spindle apparatus. Next, you need to learn about nucleus. Robert Brown discovered nucleus. Okay, and study of nucleus, that is called as karyology. Karyology is the study of nucleus. Okay, so German biologist J. Hammerling, he proved that nucleus is the controlling center of the cell, and that conclusion was based on grafting experiment by using two species of unicellular green algae. Okay, and the structure of nucleus that we can see, there are four main parts. for the nucleus. First one is nuclear membrane. Then nuclear sap or nucleoplasm. Then nucleolus and chromatin. Okay, so first nuclear membrane, we said the four main parts of nucleus. Nuclear membrane is the membrane which is enclosing the nucleoplasm. And this nuclear membrane is made up of two unit membranes and the space between two membrane that is called as perinuclear space. Okay, two unit membranes are there which is forming the nuclear membrane, and the space between the nuclear membrane, this unit membrane, that is called as perinuclear space. Okay, then comes this perinuclear space we said, and this nuclear membrane, the continuity of this nuclear membrane or nuclear envelope, that is broken by small pores, very minute pores you can see in the nuclear membrane, which is called as nuclear pores. Nuclear pores are also present. Then comes the inside this nuclear membrane, you can see nuclear sap or nucleoplasm, which is having the nucleic acids, then enzymes, lipids, minerals, proteins and all. Okay, so that is containing nucleolus and the chromatin material. Inside the nuclear membrane, you can see the nucleolus and the chromatin material. Okay, so nucleoplasm, that is the site of enzyme activities and that synthesize DNA, RNA and the ribosomal subunits you can see here. Okay, then next comes, we said inside nucleoplasm or nuclear sap, you can see nucleolus and chromatin. So next about the nucleolus. So nucleolus, that is a very dense, and it is in a spherical form, we can see. So it's a colloidal body without any limiting membrane. There is no limiting membrane for that nucleolus. Okay. So that will remain attached to the nuclear organizing chromosome. And what is the function of this nucleolus? Here, the main function of nucleolus is the synthesis of ribosomal RNA. Synthesis of ribosomal RNA or rRNA, and that is playing important role in the cell division also. Then cell division. These are the roles of nucleolus. And what about chromatin? Chromatin or that means chroma means color. So chromatin means the colored fibers. So these are the fine fibers which is formed of DNA and some proteins and some transcribing RNA. Okay, so the proteins which are associated with this chromatin, they are mainly histone proteins. The proteins which are associated with chromatin, that is histone proteins. And this histone proteins, they are positively charged and they are the basic proteins which is mainly composed of lysine and arginine. Lysine and arginine, and they are positively charged and mainly it is formed of arginine and lysine. And in some type of sperms, these histone or histone proteins are replaced with protamines. And we know that during cell division, this chromatin material will get condensed and that forms a thick structure, specialized structure, which is called as chromosomes. Okay. Chromosomes or the colored, it is like colored bodies. Chroma means color, soma means body. So it condenses and forms chromosomes during the time of cell division. Okay. So these are the main topics which we need to discuss in this chapter. We have already covered almost all the parts of 8 chapters, Cell, the Unit of Life. So here what and all we have learned? About cell, then cell theory, types of cell like prokaryotic cell, eukaryotic cell, then the differences between prokaryotic and eukaryotic cell, that we need to see all the differences while explaining about the cell organelles and all. I mentioned about the cell size, envelope, nuclear organization, DNA and histone, then plasmid, ribosome, cell organelles, microbodies, about all these, about cell wall, everything we have mentioned. Then the components of a cell like cell wall, then about function of cell wall, then the plasma membrane, function of plasma membrane, then protoplasm and that function, mitochondria and the function of mitochondria, then about plastids, different types of plastids and functions of plastids, about endoplasmic reticulum we have learned and the functions of that, then Golgi bodies, lysosome, ribosome, vacuoles, microbodies, cytoskeleton, cilia and flagella, nucleus, chromosome. About all these things we have learned. Okay. So in the next class, what we are going to do is we will go through previous year question papers. See, almost all the portions we have mentioned in this class itself. So it will be easy for you to answer the questions from each topic. Okay. Thank you.